My presentation title is Extracting Complex Trends in Gene Expression from Metabolite Data, a Cybernetic Approach. And so this is work that I did in collaboration with a number of people, including some key members in UCSD, including Manoj Maurya, Shakti Gupta, and Professor Subramaniam. And this also has been done with the help of many other people. I'd like to give a mention to Pablo Robles Granda and Mohan Gopaladesakan for their crucial contributions to this work. And I would also like to thank my PEI, Professor Ramakrishna. So before I actually get into the main idea of the presentation, I first want to just reflect briefly on metabolic systems and identify the feature of metabolic systems that we're trying to develop models to describe. And so, as you know, metabolism embodies networks of chemical reactions that work together to both break molecules such as sugars down into energy, and also to take those carbon sources and rearrange them and formulate more complex biomolecules. And so these metabolic networks, though, they're not static entities. They're in fact dynamic ones. And so for a metabolic network at some point in time, it's being monitored by some transcription factors, which then talk to certain promoters on the DNA, and those result in the transcription and translation of different genes. And so when these genes are transcribed and translated into proteins, the proteins then affect the state of the metabolic network. And so what we're trying to model is the regulation of metabolism. And so our perspective of regulation is that metabolism is regulated towards goals. And so what we would like to do is use this assumption about the nature of metabolism, that these metabolic reactions are controlled towards objectives, and then use that to predict from changes on the metabolite level what's happening at the gene expression level. And the basic idea is that proteins are, one second, this mouse keeps running away from me. So these proteins are formed in certain amounts in order to increase or decrease the rates of reaction. And the amount of protein that gets formed is proportional to the amount of mRNA transcribed from the DNA. And so by assuming that metabolic reactions are increased or decreased towards goals, we can also get a picture of whether mRNA is increasing or decreasing based off of our prediction of using cybernetic control. So I'll give you a very general idea of how, one second. And so let me first justify that idea that these metabolic systems work towards goals by talking about the concept of goal directedness. And so let's look at a very simple idea where we have two organisms at point A and then some food source for which they're competing at point B. The organism that takes the red path is endowed with a competitive advantage over the organism that takes the purple path. And so if the organism takes the red path, then it's more likely to survive. And so beyond these spatial considerations of going from point A to B, we can also think of metabolic reactions having more or less relevance towards an organism's goal. And so the cell will choose certain sets of metabolic reactions and regulate them in certain ways in an optimal fashion. And so we say this comes about through the process of evolution, which fine-tunes these organisms by iteratively selecting features that make the organisms more competitive. And we can model this using cybernetic models. So to give you a basic sense of what a cybernetic model is, we'll consider the most general definition of a cybernetic model is a set of first-order ordinary differential equations that track the time-dependent changes in metabolites and enzymes. And so the features that we include in these models are information about the stoichiometric matrix or the reaction network, and then we monitor we use a model to track the changes in different reaction fluxes. So we simplify reaction fluxes by saying that they're a function of the different network pathways and that there's some rate of uptake through each one of these pathways. And this rate of uptake is then controlled cybernetically. And so without getting too entrenched in the details, we'll move on, and then I'll show you give you a more general sense of how the model works in comparison to other metabolic models. But the other things that we include are expressions for the time-dependent changes in enzymes. Looks like some of the terms got smudged out when I converted it to a PDF. And then the important feature are these cybernetic variables. And so what cybernetic variables do is they essentially compare the return on investments for different pathways and then turn reactions up or down depending on that return on investment, which is a way of mathematically calculating each pathway's ability to achieve a certain goal. And so to give you a taste of how these cybernetic variables work, I'll give you a simple example, and then you can sort of get at least an idea of how cybernetic control works for more complicated systems. But first, let's look at this very simple scenario in which we have a cell that has two metabolic options. I'm trying to grab this arrow here, but okay. So look down here for. Can anyone see the arrow that I'm dragging around? And is the slide moving around for them too? Okay. All right, thanks. Well, anyway, so I guess I'll just describe the feature, and then you can sort of follow around with your eyes. So these two arrows represent the different pathways that we're talking about, and there are two different sugars that can be digested in this case. And so one pathway is the digestion of glucose. The other pathway is the digestion of xylose. And so glucose is a 6-carbon sugar, xylose is a 5-carbon sugar. And let's say the return on investment in this case, or this PI here at the top, is the rate at which the cell can ingest carbon. Or so the cybernetic goal is maximizing the uptake rate of carbon. And so that would be the product of the kinetic uptake rate through a certain pathway times the number of carbons specific to that sugar. So for example, with glucose, let's say we have 20 millimolars of sugar that it can digest, and an effective rate of 5 times the concentration of sugars of 100 millimoles per unit time as an uptake rate. We can then say the return on investment value for that pathway is 600 millimoles of carbon per unit time. In the same way for xylose, let's say there's a different kinetic expression for the uptake rate through that pathway, and the unregulated uptake rate then is 160 millimoles per unit time. Multiplying that times the 5 carbons in the sugar, we'll get a return on investment of 800 millimoles of carbon per unit time. So here then we can use cybernetic variables to compare the returns on investment and then determine if a reaction is turned up or a reaction is turned down based off of that objective that we've described. In this case, the maximization of the return, maximization of carbon uptake rate. And so if you compare the return on investment for pathway 1, it's 600, and the return on investment for pathway 2 is 800, we'll get a cybernetic variable value of 0.75 for pathway 1 and a cybernetic variable of 1 for pathway 2. And so what this essentially means is that the regulated rate, or R1, is equal to 0.75 times the unregulated rate. And so what essentially the cell is doing is it's turning that reaction rate down according to the objective of the cell, and that is maximizing its carbon uptake rate. So comparing this cybernetic framework with other metabolic models, we have a general description of the metabolic system here on the left side of the screen, where the arrows represent metabolic reactions and the dots represent the concentration of different chemicals. Sorry, this must be really distracting. I'm not really sure how to use the mouse on this computer. All right, so here the rate of reaction in the kinetic model, you have the rates of reaction being described as a function of the concentration of different metabolites. So if you have a high concentration of a certain metabolite, its corresponding reaction is going to be at a higher rate. The flux balance analysis approach would use linear programming to select some optimal static pathway through the network. And then on the other hand, we would have our hybrid cybernetic model describing the metabolic network as a set of pathways, as you can see here at the bottom. And then the pathways are turned up and down, represented by these little valve diagrams at the beginning of the pathways. And these valves are turned up or down based off of this cybernetic goal. So does anyone have any questions at this point about the general features of a cybernetic model? Oh, okay. All right, thank you. So cybernetic models can be used to predict a number of different phenomena. For instance, complex growth phenomena, like situations in which cells maybe consume one sugar, excrete another sugar, or excrete another product, then digest that product, excrete another product, and then digest it. They can also be used to make predictions of intracellular fluxes. In this example here, the cybernetic model can predict intracellular fluxes in Shionella onadensis better than flux balance analysis. And the other interesting thing is the prediction of multiple steady states in chemical reactors. And so the general idea is that, look at this, here we go again. The general idea is that we have this, on this plot on the left here, we can see that there's two possible states of the system for one dilution rate D here at the bottom. And so part of the power of the cybernetic model is its ability to predict unusual phenomena based off of cellular regulation of its metabolism. So moving forward, where does this presentation come in, and where does this body of work come in where we're trying to use these metabolic models to predict the time-dependent changes of gene expression? So our past models have been used to describe single cellular organisms, such as E. coli, etc. And for these systems, the objective functions are a little bit easier to formulate. For instance, maximizing the growth rate or maximizing the uptake rate of sugars is they encompass relatively intuitive metabolic goals. That is that cells are trying to compete with each other, and so if they grow faster or if they eat faster, then they're more competitive. However, when we try and shift this framework towards describing multicellular metabolism, or the metabolism of certain entities in multicellular organisms, the objective functions aren't necessarily as clear. And that's because the cells in a multicellular organism aren't competing with each other. And there's some amount of cooperation in the metabolism of individual cells in that larger organism needed so that the organism can survive. So I'll give you one example in the future, or for the rest of this presentation, where we developed a model to describe multicellular metabolism towards a certain goal, and the level, the description that we made of this system was successful. And so that specific system that we'll be talking about is the Eicosanoid metabolism system. And so these are a set of fatty acid molecules that are derived from this arachidonic acid, which have a variety of signaling functions throughout the cell. Their functions vary from the promotion of sleep to chemotaxis of different white blood cells and other things like inducing uterine contractions. However, one notable situation in which these eicosanoids participate in is the response to infection. And so that is the scenario of interest for us today for this model that I'll be describing. And so in this scenario of interest, we'll be having this cell line of macrophages derived from the common house mouse. And these cells will be exposed to a molecule called LPS, which is on the which is on the surface of different bacterial cells. And so this LPS will tell the cell, the white blood cell, or this raw macrophage, that there's an infection present in the body. And then in response to that, this macrophage will produce a number of different eicosanoids. And these eicosanoids will be produced in a certain way to then signal the formation of TNF-alpha. And so looking at the metabolic network here at the bottom of this slide, you can see LPS highlighted at the top, and that'll activate all these different signaling pathways, which will yield then the formation of arachidonic acid, or AA. And then this arachidonic acid will be further processed into a number of different molecules, including hydroxyeicosatetraenoic acids, that's a mouthful, on the left, and then prostaglandins on the right. And so the goal that we'll prescribe apoptosis. The goal that we're using to model this system is the production of TNF-alpha. And so we're saying that eicosanoid metabolism is regulated in such a way to optimize the formation of this molecule. And the reason that we chose this as a goal is that it has a fundamental role in immune cell regulation, and it's specifically well characterized for its participation in the LPS response. And so it does a number of things to fight the infection, including inducing phagocytosis and also stimulating the cardinal signs of inflammation, as well as acting as a cytokine, signaling through various pathways which are important towards responding towards infection. So the model that we'll use to describe the response to TNF-alpha is a simple linear model, and then we'll integrate this into our cybernetic model to weight certain pathways towards its production. And the way that we do this is by generating some linear model, which is which basically is a description of the concentration of TNF-alpha at any time point is a function of some linear regression coefficient, Z here, multiplied times the lipid concentration, Li. And so these Li's are the concentrations of downstream products in this network. And so basically the value of this regression coefficient Z will be in some ways proportional to the ability of the eicosanoid product to produce TNF-alpha. And so the higher that regression coefficient is, the higher the cybernetic weight will be in this variable ui. And so the cell will then turn up reactions related to the formation of this TNF-alpha and then turn down reactions that have less of a relationship to the formation of this TNF-alpha. So to develop this model, we used two different sets of data. One was time course data for the concentration of TNF-alpha, and the other data that we used to generate rate parameters for the model was the metabolite data time course. And so using just these two things, we trained all the parameters for the model, and this is all the information that goes into actually the model's prediction. So at this point, now that we have a description of the metabolic changes cybernetically, we also are calculating cybernetic control variables changing over time for these different reaction pathways. And so these, what we'll be trying to do, I don't know if this is rendering for you, hopefully it is, but what we'll be trying to do is compare the cybernetic variable U with the gene expression data. And so the cybernetic variable U is related to the induced enzyme formation in the model. And so this will turn up the induced enzyme synthesis for reactions related to TNF-alpha, and it'll turn down enzyme synthesis for reactions not related to it. So the place where we'll be comparing these variables, these cybernetic control variables with gene expression data, is where in the feature of the network where arachidonic acid divides into several different pathways. It goes through from one direction to the hydroxyeicosatetraenoic acids and then to the prostaglandins. And so for this red, gray, blue, and green pathways, we'll be getting different cybernetic variables which we'll compare to their respective genes. So to identify the genes, we went to the Kyoto Encyclopedia of Genes and Genomes to see which genes actually participated in these pathways. And a total of 11 relevant genes were identified. For the production of heat, there were a number of lipoxygenases. For the production of the prostaglandins, there were different prostaglandin synthases. And so before I actually show you the result of the gene expression prediction, I would like to make a comment. And that is to remind you that this model does not use any information from the gene expression level to make its prediction. So the only information that the model is using is the idea that, the only information is the metabolite data. And what we're saying is that those metabolites are changing to achieve certain goals, and the rates of those reactions changing are controlled in such a way that might mimic gene expression data. So one prediction we made was for the prostaglandin D2 synthase, which is here in black. The prediction is in red, and so what you can see is that both the prediction by the model and the prostaglandin D synthase, you can see that they both kind of go up, and they peak around 5 to 7 hours, and then they go down, reaching some minimal value around 20 hours into the experiment. For the genes regulating hydroxyeicosatetraenoic acid, we have the prediction here in red, and we see that the prediction shows some behavior going down, pausing around 7 hours, and then continuing to go down for the rest of the experiment. Whereas the lipoxygenase 5 genes and glutathione peroxidase 1 also show a similar behavior where they also go down. They pause in their descension around 8 hours and they continue to go down. And if one considers the possibility of delays in this phenomena, you'll also see the same qualitative behavior in the lipoxygenase 12B gene identified. So if you were to cut off the gene expression profile in black at around 5 hours, you would see the same behavior where it goes down, then pauses, and continues to go down. Also, for the pathway for prostaglandin G2, that's written incorrectly, but prostaglandin F2 alpha, you see the model predicting starting at some value, going down and then going up, and the genes go down and up, which maybe suggests, again, a possible delay mechanism here. And in fact, for all 4 pathways that we're predicting the control of, we're seeing a qualitatively similar result between our model predictions and the dynamic behavior of the gene expression. So, while maybe the prediction of one gene might not be that interesting, the prediction of all the genes as a whole is an intriguing finding. And in fact, of the 11 genes initially identified, 10 showed a qualitative similarity to our model prediction. The other prediction that this model was able to make was that for cyclooxygenase 2, which is the first step for the production of prostaglandins. And actually, all 3 prostaglandin pathways overlap. And so by looking at the control of this pathway versus that for the production of the hydroxyeicosatetraenoic acid, we see that the cybernetic prediction of this gene expression is also fairly good, where we see starting at some level, at lower level, going to a high level. And so this is interesting because cyclooxygenase 2 is very relevant to a lot of different medicines. It's inhibited in different ways by many different drugs, very well known over-the-counter drugs that do that are ibuprofen and aspirin. And so in other studies, using a model to predict the activity of this enzyme within an order of magnitude is considered a success. However, this model is going a little bit beyond that, and it's able to say that there is, it's able to describe the dynamic behavior of its gene expression. So what is the significance of all this? Cybernetic variables are used to represent gene regulation abstractly to satisfy cellular goals. And so basically, we're saying that reactions are turned up or down based off of each reaction's ability to achieve some goal. And so when we actually go and develop a model to describe the metabolic behavior of some system with this goal-based idea, we can see that the dynamic behavior of these model variables actually stimulate gene expression, which is the actual biology's way of achieving control. And so this supports our hypothesis that metabolism is goal-directed. Also of interest is the fact that we're able to predict mRNA changes from just looking at information on the metabolite level. And so given the ability of our model to make predictions, we can say that the way that we're going about modeling these systems is a correct direction to go. So what is this approach's potential? By augmenting our understanding of metabolism, using this idea that metabolic systems are regulated towards goals, we can endow ourselves with better metabolic models. And we can then use those metabolic models to do a number of different things, like better understanding cell physiology, improving metabolic properties, elucidating metabolic changes caused by disease states, and understanding why perhaps metabolism behaves a certain way given a disease state. And finally, we can look at perturbations to healthy metabolic systems or even diseased metabolic systems carried out by drugs and make better predictions of how drugs will behave on metabolic systems. So with that, I would like to acknowledge again my professor, Dr. Ramakrishna, and then all of our collaborators at UCSD, as well as a current postdoc in our group, Aravinda Mandley, and former group member, Jay Devaraj. And I would also like to thank the Center for Science of Information for their ongoing support. And I would also like to thank Mike Atwell for helping me out with this presentation. So with that, if you have any questions for the end of the presentation, I'd be happy to field them.